Okay, thank you everyone for attending, and welcome to year 3, lesson 4, protein synthesis, part 1. Okay, I'm going to now turn the time to time to Ethan, and Ethan is going to take us through the first academic principle here of the lesson. Okay, so Ethan, the mic is yours. I want you to read the concept, and then you can lead the discussion actually on this. Okay. I can help you. Yeah. In the screen, I'm showing here the presentation. Okay. Why do you think that I put these different building block sets? Yes. Have you used any of these, Ethan? Okay. It's interesting. In Argentina, I grew up with Rusty and Mecano. Okay, they are not originally from Argentina, but they are still very popular down there. And the Rusty are very similar to the Legos. Okay, you can see the shape is very similar. The concept is also very similar, that they snap, but they are absolutely incompatible. Okay. All right. So even though they are pretty close, they look pretty close, you cannot easily build things with both of them. The Mecano is actually a set that is all made of metal, and you put the pieces together with nuts and bolts. It's very, very interesting, and we used it a lot to build cars and cranes, big tall structures. Okay, really, really fun. All right, and so now let's go and see what are then the building blocks of organic compounds. What can you tell me of this, Ethan? Exactly. Yes. Yeah. So plants use carbohydrates both for energy, for storage of energy, and also for a structure. Okay, for building structures. Okay. How about lipids? Excellent. What's the main difference between carbohydrates and lipids? Excellent. Yes. Absolutely. Good job. Excellent. Okay. They are both very important and absolutely necessary. All right. But they are very different, and you hit exactly on what are the differences of each. Very good. What can you tell me here of nucleic acids? That's right. So the nucleic acids are the building blocks of the DNA molecule. Okay. Each one of these is a nucleic acid, and then when you put them together, you have DNA. What is the DNA used for? That's right. Yes. So it's the instructions of what the cells can do. Okay, that we call that the genetic code or the genetic information. Mhm. Jacob says it's as a plan for the cells. That's right. Yeah, it's the instruction booklet of life, really. Okay. What about amino acids? Did you know anything about amino acids? Yes. That's right. Okay. Here I inserted this other slide. Okay. And it tells us a little bit more about the structure here, the basic structure of each one of these building blocks. Okay. And then what they are, the names and what they are used for. All right. So, in the video I mentioned, when you see these chemical structures, okay, Haley, excellent. Yeah, the building blocks of proteins is amino acids. What type of atom is here in the intersections of the squiggly lines? Okay. What do you find in each one of those? It's another one. Okay. That is the basic element of life. Anybody else knows? Aubrey says carboxylic acid is actually just carbon. Okay. So whenever you see these lines, if it doesn't say anything in the line, okay, in the intersection of the lines, that means that there's a carbon atom. There are so many carbon joints. Yes, go ahead. Okay. So whenever you see the squiggly lines, that means here there's a carbon, there's another carbon. In this case of the fatty acids, the parenthesis here, sub N, it means that the fatty acids are long chains of carbon. Okay, so if we were to draw that, it would be, like squiggly, 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 all with just carbon. Okay. Does that make sense, Ethan? And the reason why we don't put the C, okay, is because it would be too many of them, really. Okay, so it's basically like a shorthand that people have agreed. Okay, if it's carbon and we don't put anything, it means that there's carbon there. Okay. So carbon is the main element of life. Okay, so, all right, excellent. I am muted Olson, and let me see, Justin also. Okay, so amino acids then create proteins. Fatty acids create phospholipids when they are together with a glycerol. So many times like in the case of the cell membranes, we said that the tails repel water, but the heads attract water. And that is the glycerol part. It's a sugar that is joining the molecules of fatty acids. Okay, if we see it here, this part here that has the red is the oxygen. Okay, and so that is kind of the sugar part that joins the lipids together. Okay. And so we have the nucleobases that form the DNA, and then the carbohydrates that form the polysaccharides, that is kind of the cellulose and so on. Okay. All right. So what can you tell me about this slide? I don't think that you've seen it before, but just take a time to read it, and what can you tell me about it? That's right. Uh-huh. Mm-hmm. Yeah. Mm-hmm. That. Mm-hmm. Yes. Mm-hmm. Yes. Uh-huh. That's right. So the protein, it comes really from other, especially muscle tissue. Okay. Plants also generate a lot of proteins, so there are some grains, okay, that have proteins, but you can see that there's a lot of meats that have also the proteins necessary. So what does this tell you about what you should eat? Yes, uh-huh. And the reason is because our bodies need all of that in order to be healthy. Okay. Excellent. Thank you so much, Ethan. You've done very, very well. Okay. Now I would like to invite Anna. Okay, let me unmute Anna. All right. And Anna, would you like to share with us the next part of the lesson? Yes. Uh-huh. Yeah. Mhm. Yeah. Mhm. Where is the backbone in this? What color would the backbone be in this graph? What is the backbone part? In the one on the right, this one here. Yes, exactly. Okay, so it actually, so the molecule has two backbones, one for each strand of the helix. Okay, so that this is the backbone. Why is that called the backbone? Yes. And what joins the molecules here in the backbone? Is actually this bond here, okay, that is a phosphate deoxyribose backbone, okay. Yeah. And is this join weak or strong? What do you think, Anna? These joints between the molecules in the backbone, are they weak or are they strong? That's right, yes. They are very strong, and that ensures that the molecule is not going to be easily split, divided, or, change. Okay? So, Shurana asked a question here. It says, are there always two adenine together, or can there be one adenine connected to a different type, like thymine or guanine? How do the amino parts of the amino acids, how do they connect to each other? Yes, these are the hydrogen bonds between the amino parts. If you are not using the microphone, please mute yourself, okay? So we don't have the background noise, okay? Thank you. All right, Anna? That's right, yes, okay. This is like having a book, okay, and when you print a book in a pencil, it can be erased and it can be changed. But when you print it with ink, it's a lot harder to change, okay. So in this case, the double helix is a way that God has designed in order for this information not to be easily changed, okay. It's amazing, really. Yeah. Yeah. All right. Excellent. Okay. So let's go to someone else here for the next principle. Okay. So we talk about that the how the DNA molecule is composed. I would like to invite someone to talk about the third concept of how the RNA molecule is formed and what is that used for. Caleb asked, can the DNA molecule be mutated? It can in accidental cases, okay? So it's not easily mutated. And Jacob says you could write over the ink. Yes, sometimes that has happened. And that is the problem with viruses. What viruses do is that they inject a strand of DNA into the cell, and they actually can change the DNA of the host cell, and then start producing viral RNA and proteins and everything, and that is what makes you sick. All right, that is the mechanism of how viruses attack. Okay, so how does DNA change into RNA? All right, I'm going to call on someone. Okay, Ben, do you have a microphone available? Okay. So what is the difference between DNA and RNA? What are the differences between the two types of molecules? Okay. Oh, I think that I lose everyone. Okay. Huh. Okay. Hello, hello. Okay, Anna says RNA is less stable than DNA. That's right, because RNA is just a one strand. Okay, so imagine that, and also the bond of the backbone of the RNA molecule is not as strong as the one in the DNA molecule. All right, excellent. So, Haley, I'm unmuting you. Okay. That's right. So one way to think about it is, let's suppose that the DNA molecule is the instruction manual and is inside a library, okay? And you cannot check out the books out of this library, okay? You can never check them out. So what can you do in order to go somewhere else and take the instructions with you? What do you think you can do, Haley? That's right, yes. So Jacob says you write them down. Anna says you copy down the information. And that is exactly what RNA is. So RNA is an exact copy of one side of the DNA molecule. Okay? So it's a single strand and it's a copy. Can you see that this copy here of RNA is exactly the same, has the same nucleotides as this strand of the DNA molecule. Okay, so it makes a copy, and that copy is called a transcription. A transcription is when you copy something. remember, probably you study in the times before the printing press, okay? How did people, how were people able to read the Bible? Who made copies of the Bible? Mm-hmm. Okay. So, and that Aubrey says, RNA, although copied into another chemical form, is still written in essentially the same language as it is in DNA. That's right. Both of them are made out of nucleobases, okay, of nucleotides, and they are both done in the same way. So it's still the same language. So the RNA is a copy of a gene of the DNA molecule, and then we can take that copy out of the library, okay? So the monks, in the medieval times before the printing press, people had to copy every single book by hand, and that was the process of transcribing, okay, the book. Mm-hmm. You had to make a specific and exclusive copy for you, all right? Or since other people told them when you read the book, well, yes, but if you do that, if you tell about the Bible, you are not going to make a very faithful copy of the Bible. You would have to totally memorize the whole thing, and that is very risky. And so that is why it's important to make these copies. And so the copy of RNA is made where? Where is the RNA made? Yeah, Jacob says lots of monks did that. Jacob says in the nucleus. That's right. All right, so the RNA is copied from the DNA in the nucleus, and then where does it go? Where does the RNA molecule go? Yeah. Aubrey says that there was a lot of confusion in older times when people didn't have access to the actual Bible itself. That is true. Yeah. All right. Caleb says they go to the ribosomes. Patrick says to the new cell. Not in this case. We are not talking here about reproducing the cell. We are just talking about reproducing the information in the DNA molecule. Okay? So the RNA molecule travels and goes outside of the nucleus. Let me see if I can go here. All right, so inside the nucleus we have the RNA molecule. When the RNA, the DNA molecule, when the RNA is produced, then it goes out of the nucleus into the cytosol. And there it joins, the strands of RNA joins the small protein machines called ribosomes. All right? So the RNA is transcribed from DNA, and there's another difference between DNA and RNA. One of the nuclear bases is different. Which one is that? What is another difference? So we talked that the RNA is single-stranded, okay? And Anna says uracil instead of thymine. That's right. And here you see the composition of uracil and thymine, and you can see that they are very, very similar, okay? The only difference is that thymine has this extra carbon and 3 more hydrogens. But everything else is exactly the same. So they are kind of replaceable in that sense. And for some reason, RNA uses uracil instead of thymine. All right, excellent. So that is how the RNA is copied, and then the RNA goes into the ribosome. What happens inside the ribosome? Let me see which principle. Have you had a chance to watch this video here in the lesson? This is a very, very good video and animation that shows how this happens. Okay, Caleb has another analogy. He says, it's like when you talk on the phone, the sound of your voice is translated to electrical signals that mean the same thing and can be translated back. That's right. That's right. In the case here, the reason why we call it a transcription is because we are still using the same language, okay, like Haley was mentioning, DNA and RNA, they are both still in the language of nucleic bases, okay, of nucleotides, all right? So we haven't changed the basic building block. Remember when we were talking about the building blocks between DNA and RNA, we haven't changed that yet, okay? What are ribosomes? What are ribosomes? All right, I'm going to call on someone. Emmeline, I'm going to unmute you. All right, you have a microphone. Tell me what you know about, okay, there you go. Tell me what you know about ribosomes, Emmeline. No problem. Okay. So in here in the molecule of RNA, that is basically a copy, there are several different types of RNA, and when the RNA is made out of DNA and is transferred from the nucleus to the ribosome, is called messenger RNA. Okay, and so the M means messenger. Okay, so the messenger RNA is translated now into a sequence of amino acids. Yeah. All right. Okay. Let me see. Aubrey, Aubrey has some really good comments, so I'm going to invite her to join the conversation. So I unmuted you, Aubrey. And what can you tell me about our ribosomes and how do they work? Yes. So they are everywhere in all the cells, all the types of cells. Do you know how many ribosomes could there be in one cell? I've heard that there's about thousands of ribosomes. Okay. This thing happens really fast and a lot of the times. Okay, yeah. So it is really in the thousands. All right, Patrick says that they remind him of those math in and out machines. Yes. So, Aubrey, let's see how if we can work through this together, all right? So the messenger RNA goes inside of the ribosome, and the ribosome has a special site that basically stops the RNA molecule and reads 3 nucleotides at a time, okay? So the sequence is basically broken into letters of 3 nucleotides, okay? In the lesson here we have an example, UUG, CGA, GCA, and so on, okay? How many combinations could you have if you have the 4 letters taken in groups of 3? How many combinations could you make? "64, yes. Megan, Anna, Jacob, Camilla, okay. They all say 64, 64 combinations, okay. And then there's another type of RNA that is called the tRNA. This is the transfer RNA. All right. So the transfer RNA is also a piece of RNA, okay, but has a special type of property. Do you know what those properties are for the transfer RNA? What do they do? Yes, they help decode it. So they have on one side of the molecule, let me go to the presentation and we can probably see this better. Yeah, okay. In one side of the molecule, they have also the 3 nucleotides that would match one of the letters that comes in the RNA, in the messenger RNA. Okay? So, depending on what is the combination here, only one of these transfer RNAs would match that combination. Okay? All right? So, how many types of transfer RNA do you think there are? Yes. It would also be 64, okay? You basically you have 64 different possible combinations here in the messenger RNA. So you have 64 different types of transfer RNA that would match each one of those combinations, okay? Does that make sense? What do you think that what are these little red and blue and yellow balls on the top of the transfer RNA? What do they represent? Remember that this is now we are at the molecular level, and this is all inside the cell. Okay. Jen says, I know. So they are not proteins, they are the building blocks of proteins. What are the building blocks of proteins? Yes, they are amino acids. Okay, Emily, Anna, James, Haley, they all were saying, yeah, amino acids. Okay, so yes, so they are all amino acids, and there are 22 different types of amino acids. And the type of amino acid that a particular molecule of tRNA accepts matches a particular set of nucleic bases here at the bottom of the molecule. Okay? So it's very, very interesting because if you have, let me see, if we go here, if you have the sequence guanine, uracil, guanine, is always in your RNA, is always going to match a transfer RNA that has this amino acid on the other side. Okay? So whenever you encounter this letter combination, you would have a specific type of amino acid on the other side. Okay? And so this sequence of 3 is called a codon or a short code. And the matching part of the transfer RNA is called what? What is that is called? One is a codon, okay, and what would be the thing that would match it? Mm-hmm. No problem. Yeah. So it's called an anticodon. Okay, an anticodon is the reverse code of what you read there. So for example, here in the lesson, can you see the screen? Mm-hmm. Aubrey? Okay, let me see if I can make it larger. All right. Okay, so if we have the 3 codons UUG, CGA, GCA, then the anticodon would be AAC, GCT, and CGT to match. Okay. Is anti in the sense that it actually matches. Remember that in the DNA molecule, you can only match adenine with thymine and guanine with cytosine, okay, and vice versa. And so it's the anti in the sense that that is the only way that they can be matched, okay. It's not that they are in reverse order, they are the mirror image of them, okay. All right. Mm-hmm. Yeah. So, and then once they get here once the transfer RNA matches the particular, let me go to the presentation. Once the codon is matched with a particular transfer RNA with that same anticodon, then the amino acid is released inside the ribosome and is appended into a strand of amino acids. Okay? And then the molecule is advanced once one nuclear, one set of one codon more, the empty transfer RNA is released out into the cytosol, and then the next one comes in. And the next amino acid would be joined into the chain of amino acids. Okay? Does that make sense? Mm-hmm. Anna asks, what happens to the tRNA? What happens to the transfer RNA once it releases the amino acid and it goes back into the cytosol, into the cytoplasm? What happens to the transfer RNA? What do you think? Anyone? What happens to the transfer RNA? So here we have transfer RNA that is charged with amino acids, okay? And then once it releases the amino acid, it goes back and is not charged anymore. So the protein is this sequence of amino acids that is starting to grow. Okay. Emmeline says, is it considered waste? Anna says, is it recycle? Okay. What do you think that will happen? It's very strange that things are usually not wasted in nature. Okay, everything is used to the maximum. Okay. So what happens with the transfer RNA, once it has released the amino acid, it goes back and seeks another amino acid of the same type. Okay, so remember that there are amino acids, free amino acids all over the cell. Okay, we eat a piece of meat, our stomach digested that and breaks down the proteins from the meat, the hamburger or so on, okay, and breaks it down into amino acids. The little amino acids then go through the blood, and from the blood they enter into the cells, and they are floating around the cells until an empty transfer RNA catches them, okay, and then puts them into the ribosome and puts them to work in part of the protein. Does that make sense? How it goes? Yeah, Camila says it is released to attract another amino acid. Okay. Yeah, it's extremely cool. It's really, really amazing. So these transfer RNAs are reused all the time, and it just keeps going because the body needs continuously to build different types of proteins. Okay, that is why we continuously feed things. If a protein is no longer used, then the protein goes into the lysosome, and the lysosome has other proteins there that break down the chain of amino acids, and then they release the free amino acids back into the cell to be recycled and made into a different protein, whatever the next instructions coming from the RNA are. Okay. That's right, yes. Okay. So for example, if you exercise a lot, all right, your body is going to receive signals that it needs to build proteins that are called actin proteins to build muscle tissue. Okay? And so the DNA, let me go to the presentation here. The DNA molecule that has the gene for the actin protein is going to be read. Is going to produce a strand of RNA that has the genetic information of the actin protein, the protein for muscles. Then that is going to go to the ribosome and is going to start producing actin proteins all around. Okay. This strand of messenger RNA, when it's read, it can be captured by another ribosome and read again and make another actin protein. It can only make the same type of protein because it has the instructions for that specific protein. Okay, but it happens thousands of times. All right, very fast also. So the process now of changing the message of RNA into the sequence of amino acids is called translation. It's called translation because now we are changing the language. We are changing the type of building blocks. Okay, so the body cannot use nucleic bases for making structures. It can only use it to transfer the genetic information. But the proteins are very, very versatile, and they can be used for a ton of different things. And so that is how the body translates the genetic information into RNA, and then the RNA is made into proteins. Okay, let me see. Jens says, so in the word of wisdom it says that we should eat meat sparingly, and amino acids come from meats. Is it unhealthy to have too much amino acids? Well, so amino acids come from meat, okay, but also there are many, let me see if I go to the, okay, here, there are many vegetable sources of amino acids also, of proteins, like nuts and seeds. A lot of the fruits and vegetables also have proteins. So meat is not the only source for amino acids, but is a rich source of amino acids, but also milk and other vegetables have also proteins that when we eat, we can break down into the amino acids that we need. Emmeline here asked a question. I'm going to re-ask a question. I have a little bug. When our amino acids begin to be unbalanced, is it due to our lack of eating nutrients, or is something wrong with the ribosome or our RNA transcript? Okay, this is a very good question. Most of the time, if we have an imbalance in the type of amino acids that we get, is because of an unbalanced diet. Okay, and so we need to be careful to have a balanced diet in order to obtain the nutrients that are needed for the body. Okay, so like even fats, a lot of people say, well, eating fats is bad. It's not bad because, like we were talking before, all the cell membranes, most of the brain is made out of fat. So if we don't eat that, we are going to have an unbalanced diet. There are a very, very few people that sometimes have genetic problems that cannot metabolize things properly, and so they have an imbalance in their nutrition. Okay, and so then they have to compensate for that. All right. So here, so Caleb says it's the way the fat is stored that is the problem. That's right. So if you have too much of a good thing, it can be too much. All right. Most of the times, the things that are the molecules that are water soluble, if the body has too much of them, then they can excrete in sweat or urine. All right. But if you have too much sugars or too many fats, the sugars are transformed into fats, okay, and they are stored. and the body usually stores as much as possible, and so you need to be careful not to eat too much of those things. Jens says it's cool how many things proteins can be used for. So yes, proteins are really the building blocks of almost everything in the body. Okay, they can be used for lots of different amazing machines. So here I want you to meet the amino acid, the building block of that, okay, of the proteins. And as you can see, all the amino acids are made out of the same base, okay, an amine part and a carboxylic area, okay, and then this R means the residue, which is kind of the tail of the different amino acids. This table has the 22 basic amino acids, and as you can see, each one is slightly different than the other. Some are positively charged, some are negatively charged, some have a loop of carbon, okay, some have sulfur, all right, in them, some have nitrogen, okay, and so depending on the sequence of the RNA molecule, we are going to basically build this sequence of amino acids, and then they are going to join, the amino acids join one side, okay, of one amino acid joins with the other one, and this part is going to join together and is going to form what is called a peptide bond. This is a strong bond between the carbon and the nitrogen, and is going to release one molecule of water. And so that is how the amino acids are joined together in this long chain that is going to make the protein. Let me catch up a little bit. Aubrey says, under eating vegetables can affect people usually, and if you if too many amino acids, it will get eliminated, I believe. Yes, so there are some proteins that are essential to the human body, okay, and that we cannot create on our own. And so those proteins are what we call vitamins. So the vitamins are specific types of proteins or substances that our body needs, and it doesn't need sometimes a whole lot, but you need at least a little bit in order to create the things that are necessary for life, okay? And those are the vitamins. So vitamin C, vitamin B, vitamin E, and so on. They are those specific things that are needed to create these machines. Aubrey says, so there are about 22 amino acids that your body needs. Does that mean all the other ones don't have protein? So what happens is that we are going to talk more about this next class. Okay, that is why this is the protein part 1. But next class we are going to talk more about what happens after this chain of amino acids is put together. What I'm just going to tell you is basically that the chain of amino acids starts folding, making structures, they combine, and then they make beautiful and amazing biological machines. Okay? All right? So, Patrick, I would like you very briefly, because we don't have, sorry, I went out of overtime, but I want you to talk some about the gospel principles that we had here for this lesson. Okay? Mm-hmm. Yes. Mm-hmm. Excellent. Mm-hmm. Yeah. Mm-hmm. Mm-hmm. Yeah, I have a question. The four principles and ordinances of the gospel, are those things that you just do once and you don't need anymore? That's right, yeah. Do we need faith in Jesus Christ just when we are converted? No. Yeah, how about repentance? Uh-huh, we need it all the time. Well, but sorry? Uh-huh. We are baptized only once for ourselves, okay? But how do we kind of get rebaptized all the time? Through the sacrament. That's right. Yeah. Okay. And receiving the gift of the Holy Ghost, how do we maintain that going? Yes. Uh-huh. Yeah. Mm-hmm. So Caleb says principles are all the time, ordinances are once. Yes, we do the ordinances once for ourselves, but we need to keep the Holy Ghost with us all the time and basically be purified as in baptism also every time. Mhm. Okay. How about the second gospel principle that I have here? What can you tell me about that, Patrick? Hmm. Hmm. Yes. Hmm. Excellent. So he's making a copy. Hmm. Uh-huh. That's right. Yes. Uh-huh. How about in the Book of Mormon? Hmm. Yeah. Okay. Okay. So remember when Nephi copies the Isaiah chapters in the Book of Mormon? Where was he copying them from, and where was he copying them to? Uh-huh. Okay. Uh-huh. All right. Anyone? Mm-hmm. Okay. Uh-huh. So Michaela says the brass plates to the second book of Nephi. So Nephi transcribed the Isaiah chapters from the brass plates, and he just copied them into his plates. Okay, what later on became the second book of Nephi. Okay, so it was just a transcription there. All right. The other example that you talk about when I think that it was King Mosiah, Limhi's people, they found the Jaredite plates and King Mosiah got them. He actually translated them into the Nephite language. Okay. So does that make sense? Okay. All right. Excellent. Excellent. So we also have examples of transcription and translation of the scriptures, okay, and of the Word of God. Mm-hmm. Similar to how it happens within the body. Okay, excellent. So I want to tell you guys, thank you, Patrick, very, very much for that. And I want to tell you, the more I learn about these processes and all these things, the more amazed and awe I am at seeing the hand of God in His creation. It is just incredible, really, what He has done for us, isn't it? And I want to testify to you that this is really the hand of God. It's not something that has happened by chance or anything remotely that way. Okay. And I would like now to invite Jens to say the closing prayer for us. Okay. Jens? Anna says it is so intricate and amazing it couldn't have happened by chance. There's really not a chance of that. Okay. Okay. Yes, we can hear you. Mm-hmm. Thank you, Jens. Mm-hmm. Amen. Thank you very, very much. Yes,